Okay. Do you want to go ahead and welcome people?
Yeah, everyone, I want to thank you for joining us in this webinar. This is the second year that we will be funding both translational science and translational research projects. We've had very strong responses before. We've had outstanding projects. The projects are metriced to lead to independent federal funding and or high impact journals. So our goal today for Arnold and I is to assure you we want you to be competitive. We are interested not in the exercise of writing grants, but in the capability of you all to use the funding for your own career development and for projects that will lead to high impact. So with that, I'm going to introduce Arnold Rabson. He is one of the co-leads and has been running the pilot program. I can't believe it. It's going to be coming up on its seventh year, Arnold. Time flies. I'm Ray Panettiere. I am the program director for NJACS, the New Jersey Alliance for Clinical and Translational Science. This is the only CTSA hub in New Jersey. And we're thrilled to also work with our alliance partners, which is Princeton, NJIT, Rutgers, and the Robert Wood Johnson Barnabas Health System. So, on behalf of all of the alliance partners, all of the administrators that work to pull this together, we welcome you to this webinar. All right, Arnold.
Perfect. I'm going to go ahead and share slides. Let's see if we can make this work. Let me see if I can do it just with PowerPoint. Okay, and now let me know if you're seeing it as
Excellent.
slide
Yep,
presentation.
you got
Okay,
it.
we'll see if I can make this work. Okay, welcome everyone. I echo Ray's welcome and the sentiments that the goal of this and of really everything we're doing is to try to have the best possible science be funded and move forward to advance translational research, collaborations, and translational science and research collaborations at the institution, and hopefully to help everyone understand better what the different mechanisms are and how to move forward to be successful in those mechanisms. So again, welcome. Let's see now. How am I gonna advance this? My advance button isn't working.
I think
Let's see.
there you
That
go.
that
It just
advanced.
takes a
I'm not
little
sure what I
time.
did. I'm not sure what I did, but I'll see if it works next time. I think I may have skipped one too.
Yeah, you need to just go
There
back.
we go.
There
Okay,
you
so
go.
now maybe it's going to work. Ray kindly introduced me. I'm at the Child Health Institute of New Jersey down at Robert Wood Johnson Medical School, and the co-leads include Samuel Wong from Princeton, a neuroscientist of great renown who has also been a co-lead for a number of years now for the pilot program. And more recently, Jonathan Grassman joining us from bioengineering at NJIT. And we've had a really great partnership with NJIT through the years. And we welcome Jonathan and his work in the area of vascular and neuronal innervation and sort of tissue construction, reconstruction, which fits very nicely with a number of the sorts of areas of excellence here and the themes of NJIT that are brought to the CTSA, to NJ Acts. And as well, Joseph Jager, I don't know if I pronounced it right, who's a doctor of public health, master of public health, and the chief academic officer at Monmouth and the associate vice president for research at RWJS Barnabas, really emphasizing how important our collaboration with the hospital system is, that they're really a full participant in this process. So we welcome them, and we're all here to help and to make sure this process works well, works fairly, and works to help you develop and present best applications possible, and then to fund the best possible science. Um, these are really the most important people in this process as the pilot program administrative staff, and I will call out Cassandra Burrows, Sandy Burrows, who's on the call as well, and Anthony Gonzalez as well, who really are sort of day-to-day leaders of a lot of this. Pam Dolan really helps make the whole submissions process move forward. Sandy is involved in making sure that the right funding is done in the right way. And Anthony has been just a godsend helping navigate through the regulatory compliance and really putting together submissions and, at each level, a letter of intent, et cetera. So deeply grateful to them, as well as to Judy, Bill, and Elizabeth from Princeton, who provide just invaluable support, advice, et cetera. So our goals today, as Ray said, is to really introduce this sort of bifurcated or two-armed pilot program system. And we'll talk a lot about the similarities, differences, and the how-tos related to the clinical and translational science pilots, as opposed to the tri-institutional clinical translational research pilots. And we'll spend some time, and oops, sorry about that, some time, and I urge you to as well on your own, talking about the difference between clinical translational science and clinical and translational research. And this is something that I certainly had to be educated on and have a better and better appreciation of now. And we'll talk about it. We'll talk about examples, but I also would refer you to the website that you'll see later from NIH, from NCATS, that provides more examples, etc. Because it's an important but a significant difference between the two that can get a bit confusing, but I think as one lives with it a bit, it becomes clearer. And obviously, we're here to answer questions so you can develop letters of intent and move forward with the process. So now we're again the two processes, the Translational Science Initiative, Clinical Translational Science, and Clinical Translational Research, have a number of important similarities. They both have a two-phase application process involving a letter of intent, which will be evaluated, as we'll describe below, most importantly for responsiveness to the program for which they are submitted, and also for competitiveness on it. To be honest, there needs to be that because we get large volumes of applications and a lot of work to do in the review. And also then solicitation from that set of a full application. So letter of intent, following sort of approval of the letter of intent, solicitation of the full application. We'll go over some of the application requirements, most of which are really the same. There are a little bit of difference in PI eligibility we'll mention. We'll talk about the review criteria and processes for each, the deadlines, and how the regulatory requirements are handled with respect to each of them. And then reporting requirements are very similar, and that's outlined in the RFA. We won't spend a lot of time on that today. Let's see, I just want to make sure I didn't miss. Okay, so here we go. What's the main difference between clinical and translational science versus clinical and translational research? And what I'd say as we get into this is many of us were brought up on the concept of clinical and translational research, and we'll talk about that, but the evolution of the concept of clinical and translational science and what it does to move translation along is something that I certainly have been learning and appreciating more and more as we go on. And I think even for someone like Ray with a long history in this business, this has been sort of an understanding of how to develop this newer sort of scientific approach to making translation work better. So translational science, as opposed to translational research, and you can read it, but I'll read some of it, is focused on understanding the scientific and operational principles underlying steps of the translational process. And I will highlight again this website, and perhaps, I don't know if we're sending out slides, or we could perhaps put in the chat, Sandy or Anthony, the website, the link. Oops, excuse me. And so that people can go directly to it, because I do recommend going to it. It does elaborate more than we'll talk about these points. But the basic principle of translational science then is to catalyze innovations to overcome challenges, long-standing challenges, along the translational research pipeline. So we'll talk about the translational research pipeline, the classic sort of basic, be it bench, be it population sort of research, utilization research, but to go from there into clinics and into populations as the pipeline. Again, we'll go through it more, but there are disease agnostic barriers that come up in these sorts of translational activities. And the goal of translational science is to study what's causing these barriers and, more importantly, to develop innovations to overcome these sorts of barriers. And these barriers can be scientific, we'll give examples, operational, financial, administrative, and to really develop the innovations in these areas to transform the way that research is done, making it faster, more efficient, and more impactful. And Ray, I would say again, interrupt me anything you want to
Yeah,
elaborate.
no, I'm going to interrupt on the next slide.
Oh, perfect. Great, great. I talk fast, so I want you to make sure you get
No.
in there and interrupt. So yeah, I can let you interrupt on that.
All right, so let me take this one, Arnold.
Mhm.
So the important issue when you're thinking about translational science is that you're studying process. There's a couple catchwords that we're looking for as well as NCATs. Now, this component of the BiFed grant system that we have is funded by NIH. That means it has to adhere to all the NIH priorities and the regulation and regulatory processes that are necessary. So we will determine who is which is the best meritorious grant. We could then recommend funding. It has to be approved in part through NCATS, which means A, it has to reach the NIH priorities. B, those priorities need to be aligned. We are all familiar with new terms that we shouldn't be using. We should be looking at public health, basic science, preclinical research, clinical implementation, all geared towards patient involvement. That's what translational science is. Now, that sounds like a lot of words. Panettiere, tell me what I need to know. Well, there's 5 grants that will be awarded. Each of these grants are at 25,000, and it's for a year. So make sure that you recognize scope. We're looking for grants that can utilize that, pretty small quantity of money to jumpstart their pathway to more substantial success. The next series of grants that we'll be talking about, the translational research grants, are more substantial. They're 50 to 100,000, and they could be for a year with an extension for another year, not of added funding, but to spend out. But the translational science grants are for 12 months. They have to start and finish in that same framework, including all the regulatory processes that we and NCATS needs to approve. So what would not be competitive in translational science? If you're focused exclusively on one disease state, that may not be competitive. What we're looking at is a study of process to overcome hurdles and chasms in the pipeline. So, for example, you want to compare pathway A and pathway B. Which of these pathways could overpass or undercut a fundamental chasm in our approach, the study of process? Now, it could be more basic research where you're going to be doing, let's say, screening of molecules for therapies, and I'm going to compare pathway A and pathway B. Recognize it's $25,000 and it's got to be done in 12 months. All right. So you're not going to be doing a lot of breeding of mice and things like that. And you're studying the process not necessarily the outcome of a disease state relevant outcome. You want to compare pathway A and pathway B. I'll give you an example. Now, this was a bigger scope grant, but there was an unmet need in our health system where we don't get patients to see medical geneticists in a timely way. It takes about 9 months to see or get a referral to a medical geneticist. So these investigators studied a process of what if we used genetic counselors as a very quick way of getting an evaluation compared to the standard of care, waiting for the medical geneticist evaluation, and looked at quality, time to diagnosis, number of patients who could go through this process. And what was found is a geneticist, a medical a genetic counselor could do this fold faster than waiting for the medical geneticist with an improvement in diagnosis. So I use that as an example. Pathway A, pathway B, you tested it, and you came up with an outcome that was measurable. So it was simply one example. It is complicated, so I do ask you to go back to that web link and take a look at what translational science is. It would be helpful in your application to have two components. Number one, what is the roadblock? What is the roadblock you're trying to surpass? And the second thing that would be helpful in the application is how are you going to leverage the services of NJX to accomplish the goal? Things that are going to be not competitive for translational science pilots are a very, very specific outcome in one disease. Remember, this has to be disease agnostic, scalable, and generalizable to other therapeutic areas. That's number one. Number two, don't propose a clinical trial that's going to be a 12-month project for $25,000. It just ain't going to happen. So these are things that are clearly not going to be competitive. You'll put it in your letter of intent. Our committee, executive committee, goes over each and every one very carefully. We deem it going to meet the needs, and we can move forward. So next slide.
There we go.
There we go. These are the bottlenecks in translational science that you want to overcome. They are basic clinical research and dissemination and implementation. Target qualification, I'm not going to read all of these, but these are absolutely aligned with the translational science pilot programs, where you're going to look at how to improve process. Each of these bottlenecks causes a dramatic decrease in the flow of new products, new approaches, new applications. Remember what I suggested, identify the bottleneck, and remember, we're studying a process that's scalable and could be generalizable. If you can build that in your LOI and then your application, you'll be competitive. Remember scope. Scope is not a lot of money, and it's got to be done in 1 year. Next slide. Re-engineering this translational pipeline is what NCATS wants to do. If we can remove the bottlenecks, we're going to go from molecule to changing policy much swifter. That was one of the major tenets and priorities for NCATS. In our application, we had to expound on this extensively. The good news is we were refunded for 7 years. We're in our second of the 7 years now. So I'm sure you're going to hear about this over and over again. But taking an examination of your project within this conceptual framework is very important. Eliminating bottlenecks, disease agnostic approach, scalable, generalizable, and within the context of the limited funding and support we have. This grant, that is the translational pilots, will be $25,000. We're looking to fund 5. Next slide. So this is at what's not and what is translational science. I think we covered these quite well. Looking at the left side, just picking one, evaluating the toxicity of a newly developed compound to treat Alzheimer's. That's going to be very disease specific. It doesn't study process. If you're developing a new assay based on human cells that can identify potential toxicities across diseases, that's the critical term, that would be compliant and would definitely meet the goals. Another, systemically studying barriers to enrollment in historically underrepresented groups in clinical trials. Maybe you developed, and this was a successful project last year, kiosks that was 24/7 running that allowed patients to access any clinical trials in the Barnabas Health System by scanning a QR code on their phone. That was compared to a traditional approach. The outcome was number of patients number of participants in clinical trials. So notice the difference. You could say, well, it's nuanced planetary, but it isn't. You're studying a new process, a kiosk, scanning with a phone, and access to any clinical trial is vastly different than looking at just a clinical trial based on breast cancer on the left side. So I tried to be very, very granular in this approach. You'll be able to pull this up on the internet as we have this put together. And I think you'll get the flavor for science versus research. This is the component that is translational science. We're going to be talking about translational research in a minute. Next slide. So these are examples of the NJX Clinical Translational Science awardees of last year. There was a grant given for testing a platform for probing the role of dendritic structural plasticity in neuropsychiatric diseases, not just schizophrenia, major mental illness, ultrasound responsive therapeutic gas microbubbles for the localized treatment of cardiac injury. This sounds a little bit more disease specific, but embedded in this application was the use of microbubbles in a variety of diseases. They used cardiac injury as simply the example. Preconception care and also prevention and identifying underserved populations for getting preconception care, and then assessing the engagement, as I mentioned, of patients using kiosk videos. So these were these were 4 projects that were funded last year. Next slide.
The next slide's going to be some more, and I'll just, before I do that, just emphasize what Ray said about the second one. If you're writing an application for science, translational sciences that has a more focused piece to it, make sure you're showing the generalizability of it. And really spend time on that.
These are other ones. I'm not going to read each and every one. They will be accessible, but a rapid low-cost SNP detection with occluded Cas13. The last one, an optical contrast agent to enable in vivo imaging of immune cell activation in response to immunotherapy. Very general. This was disease agnostic immunotherapy and imaging of immune cells, but very focused on immune cells. So you can have very specific projects, but you need to fan it out across a variety of disease states. Next slide.
And I'll highlight the bridging decades, the data architecture across sort of historical cohorts. That was a very interesting, important process. a grant, really the idea of linking different data sets that have been difficult to link before to solve problems in translation. So that I think got a lot of support as well.
So the clinical and translational science projects last year were $40,000, addresses how we improve process that facilitate and support research, test two or more approaches to solving challenges, can be hypothesis driven. That is, pathway A is better than pathway B. Yet not maybe the same kind of hypotheses you write for an R01 if you're a fundamental scientist, but you understand you can study differing or parallel pathways to get to an outcome. You identify, as I mentioned, really important, a roadblock for translational research and demonstrate the relevance for others doing research. Okay.
And I would stress again what Ray said, the is hypothesis driven. The point there is it is science. It will not be reviewed if it's just sort of, well, we're going to put together a new program.
Yeah.
We're going to put together a new program and test how it effectively addresses a roadblock.
In comparison to another project, right?
Right.
Great. Okay. Projects should focus on challenges, roadblocks, approaches, and process to address them, be disease agnostic, and align with one of the themes. The themes are there on improving outcomes in translational science. We have had at times projects that were education based. Can we improve
Sorry, I'm trying. I'm
training?
going the wrong way. I'm sorry.
That's all right. Can we improve training? That would be competitive if it's pathway A and pathway B. So next slide. And next slide.
Sorry. Yeah, yeah.
Expanding patient and community engagement, certainly a valuable endeavor for NJX. We have an entire module within an element based on patient and community engagement. How do I bring the community to science in a bidirectional way? Tell me how. Come up with a new process, comparing process A to conventional process. You could implement evidence-informed practices for patient and community engaged research. And an example might be testing novel recruitment strategies, pathway A, pathway B, and studying the process. Next slide. Bold scientific approaches, ambitious research goals are fine, but they must be within the constraints of the funding. I failed to mention, I said 25,000, it's 40,000. We want to make sure that in that 12 months with $40,000, you can get the work done. There is no carryover for NIH money. It has to be spent within 12 months. So if you need IACUC approval, if you need IRB approval, that's going to burn daylight. You don't get an extension after 12 months. What you don't use, you lose. Research priority setting, diverse perspectives in research through partnerships. We like to hear that you're going to be using NJX services. Remember, those services are free of cost. We have a very, very accomplished and expert biostatistics epi group that could help you put together your data analysis, and you're studying research processes and structures. Next slide. Training and education. We have workforce development. How do we build a better workforce? Make sure you mention rigor and reproducibility, create novel training programs. Regulatory is one of our cores. They can help you with submissions of IRB. Anthony Gonzalez is on here. He's a wonderful resource. He's also an expert in REDCap and questionnaire generation. These are free of cost services. Building them into your proposal and suggesting you're going to use these services is a strength. Next slide.
I just say, because we've seen it before in sort of training programs, education, if you're developing in this area, make sure you describe how you're going to evaluate the effectiveness of what you're doing. It's that first line that's very important.
Yeah, metrics of success. All right. So maybe Arnold, you could take us
Sure.
through this piece.
So this is the more traditional sort of thought process around translational research that many of you have heard about for years, which is truly bidirectional, but classically translating discoveries from the laboratory to bedside to populations. But it can go the other way as well, identifying important bedside sort of piece going to the lab and then back to the bedside or to populations. So this is what, again, many of us were sort of trained with growing up and can allow much more focus in terms of the disease, that you're looking at, et cetera. Again, it's better though from the trans standpoint of NJACs and of NCATs, that whatever you're doing has an implication of potential generalizability. But again, translational research means moving forward and can involve more specific sort of diseases. Is that a fair statement, Ray?
Oh, yeah, absolutely. It's spot on. I think most of the people on this call who write R21s, R01s, and NSF grants are very familiar with the traditional translational research. This could be going from a mouse to a human in a rigorous and testable manner.
Yeah, or testing a new assay that might predict prognosis, et cetera. The kinds of things we're used to in that way. Translational research, as I'm sure you're aware, again, is a continuum, and the idea of developing new approaches, the transfer of applying discoveries from basic science in different ways, basic research in different ways, and that again can be social science into populations as well as biochemistry into patients. It really basic research sort of spans that in this endeavor, and demonstrate usefulness through clinical trials and studies, and disseminate findings to enhance best practices. So into the community and in impacting policy. So really a continuum here with feedback between different components of the continuum. Now this is very important. Pilot proposals must demonstrate clinical relevance. So, a fully basic science project without really showing what the clinical importance is not going to score as well. There are venues for funding that, but here we are at NJACs. It is clinical. It is the translational research component here. So take enough time to make sure that you're really outlining, just like for any grant, outlining, walking the reviewer through what the clinical relevance is. Don't leave it to the imagination, or don't say, oh, everybody knows that this is, that these enzymes are important for heart disease.
Yeah, so if I could add, Arnold,
Yeah,
so what
please.
would not be appropriate? You're a zebrafish scientist, and your first sentence is clinical relevance, and your last sentence is clinical relevance, and you go into depth. In between all of that, the zebrafish modulating function, physiology, pharmacology, that's not going to work. You're going to have to weave in throughout your proposal the clinical relevance. Best way to do that sometimes is to use organoids, human tissue, human cells in conjunction with more simple manners or models and platforms.
Thank you, Ray. And just to say about this, here there's a sort of fixed number of dollars that's available, but in the past there has been some flexibility, and I suspect will be again in terms of the actual award depending on the number and the quality of proposals. There is a sense there has to be enough money awarded to be able to do a project, but on the other hand, we've had some outstanding projects in the past and have felt it more important to spread it out and make sure we hit a one extra, for example. So there will be some flexibility here based on of number, based on the quality and number of proposals. Collaboration across the NJACs institutes is strongly encouraged. It's not required, but there can be real advantages to bringing together expertise from across the institutions to get what you want to get done and to demonstrate building a team that will have legs, that will take this project forward. Again, these are pilot proposals. And as we'll talk about in a moment, as they're being reviewed, one of the considerations is how likely are these proposals to generate long-term, larger kinds of funding. And again, collaboration, bringing in all of the right sort of expertise, potentially across the institutions is really a valuable thing to be doing. The application types, there are three major types. The first is the clinical and translational research projects, and I suspect, having looked at the names, a number of you have applied to some of these before. And these really can be anywhere along the entire range of the continuum. Sorry, that's my um, ah, here I go again. Let's see. I'm sorry, I'm have a very sensitive mouse, and so the clinical and translational research projects across the continuum. So they can be laboratory based, clinically based, health services, epidemiology. You can read it, engagement, diversity research. Again, we all have to understand some of the changes in funding priorities across NIH now. So in fact, we haven't changed our slides. But the idea of diversity in this setting, one of the things that we didn't stress at the beginning, but from the beginning of the grant, Ray has really stressed heterogeneity of clinical presentations, clinical populations, approaches to therapy. So, in a sense, diversity also means heterogeneity here. And I think that's a really important point that's very consistent with the CTSA grant and the theme of this particular CTSA. So here we go. That's the next line. I'm sorry again. Um, Oh man, what have I done? I keep going backwards and then it gets stuck.
There you go.
Projects that focus on heterogeneity of disease and heterogeneity of response to therapy. How do you move towards more individualized therapy or encouraged? So the Propel projects, in sort of a very crude analogy, the first ones would be more like your RO1s, your multi-PI RO1 sort of projects, probably smaller than program projects, but again, a little bit bigger. The Propel projects are more like the high risk, high reward sort of R21-ish projects. So taking a little more of a risk, catalyzing discovery through proposals that look at new ideas, new technologies, bringing new methods to a particular project, a particular area, enabling clinical data acquisition, small equipment, licenses to data sets that can really move a project along without necessarily the sort of strength of background, and preliminary data, et cetera, that might tend to be more a part of a really well-knit clinical and translational research project. Then the third mechanism that we've supported over the years is called the valued partner projects. And these support team research and importantly, build liaisons with key partners outside of our CTSA group. So key partners in industry, in government, in nonprofits, other CTAs would count, or insurance companies, for example, through data sets. The partner has to provide support, and that has to be documented. So a detailed letter description of the support. It can be in-kind services, access to databases and early analysis to get you to what you need, or some sort of funding to help with accessing a population, for example. And so there needs to be that detailed description and the letter of support from the partner organization.
And it would be, it is very reasonable, colleagues, if you want to monetize that in-kind support and state it unequivocally. This license typically costs X. This company is going to volunteer to give us access, and that would give the reviewer a sense of the value of that interaction. So no reason to be shy or bashful. We want to know what is the ask and are you going to receive if funded?
Absolutely. Absolutely. Again, some of the projects awarded over the last 2 years, you can see them. We don't need to look at all of them, but microfluidics, new sort of microfluidics or in vivo testing, quantifying change readiness and psychotherapy outcomes, the effects a couple of years ago, the wildfires and the particulate matter, youth perspectives on positive childhood experiences and looking at community interactions and assets, neuropathic basis for dental pain, a clinical pilot. So as some of the examples, and see, I think we only had the 1 year there. Um, so the next slide, and again, I believe you will be getting the slide set will be available as well. It really details some of the differences and similarities beyond what's clinical translational science versus research. There are some specific differences. Funded through the CTSA, investigators sort of leading projects from all four partners are eligible for the CTS. Because of some the way the funds have flowed from the individual institutions, which are the contributors for this clinical translational research, or the three institutions, Rutgers, NJIT, and Princeton. Because of some of the fund flow recently with Princeton this year, eligible leads for the CTR will be from Rutgers and NJIT. CTSA award we just talked about from NIH NCATS, whereas money from Rutgers, Princeton, and NJIT institutional funds are going for the clinical translational research. An important point Ray really stressed is that the NIH funded, NCATS funded CTS awards, the regulatory approvals have to go through NCATS. So if you get this award, you need to move things as fast as you can because there is a delay sometimes in that. There have to be the regulatory approvals, obviously, for the CTR, but it doesn't have to take the diversion down to Bethesda and back. And I don't know how NCATS is doing, Ray, but there are other parts of Bethesda where they've lost some people in the support staff and things can take a little longer. Um,
So, how can you get anything done, right? Is one of the
yeah,
questions people say. Well, the best way is to have an IRB approved
yeah.
protocol before you're going in because that takes that off the table. It's there, it's ready to roll. Same thing with IACUC. If you have the IACUC protocol already there and it's approved, that will make things go much swifter. If you're going to wait and find out if you got funded and then apply, you're going to be 2, 3 months potentially behind the eight ball. So that would not be something beneficial for the CTS projects.
Absolutely. I'd second that. And if you don't have it now, maybe in the course of preparing the application, you also get the submission done, for example, that sort of approach. Um, Clinical trials really are not, neither is geared up for supporting direct clinical trials. We talked a little bit about approximately 40,000 for the CTS, 4 to 6 awards. We talked about 5 before. 2 to 3 awards, direct costs only of $50 to $100,000 for the set of the CTR, the clinical translational research projects, Propel, and the valued partner. And again, there may be some flexibility in actual amount, depending on which come in which mechanisms and sort of what budgets look like. Again, just what we were talking about. The CTS, 12 months or less, depending on the NCATS approval date. So the more you can do ahead of time, the better. 12 months for the CTR. Again, start dates for both are going to depend on getting through the regulatory approval with the NCATS piece. And the NIH, no cost, no extension allowed. We are allowing a 12-month no cost additional extension for the CTR.
A
Um,
couple comments
yeah,
I'd like to
please,
make.
please.
Uh, be very cautious about proposing anything with any kind of foreign component at all, be that a COI, be that anyone or participants in studies that are from a foreign outside of the US. The current priorities of NIH is not to embrace foreign research. So I'd be very cautious. We've had issues with this. It gets brought up not at our level, but at NIH level, and could disqualify. There was a question in the chat regarding the use of human tissue. to predict, develop, validate, or improve diagnostics. That could fit on either side, depending on scope. So I think the fact that you're using human cells derived, if those cells are available, the tissue's available right now and de-identified, that would make things go smoother. Really depends on the regulatory approvals that are necessary. Sorry, Arnold, go
No,
ahead.
I think that's great. Thank you. I can't see the chat because I
Yeah,
tried
no,
once, but then everyone sees my chat and some of it's directed to me. So I won't. So please do keep an eye. Thank you. And interrupt, as I say, every single one of these important points. Eligibility, well, like all grants, be well suited to the project. Have complementarity from co-investigators if there's aspects of it that you really are not as well suited for, but there are other people here. You, like all grants, need to be able to devote sufficient effort to do the aims. Again, just like the first aim, appropriate experience and training to lead the project, ongoing accomplishments in the field. Um, if a project's collaborative, investigators, again, complementary, integrated, and the PI needs to have a primary affiliation with the alliance partners. And Rutgers, Princeton, NJIT, or Barnabas. And we talked about some of the specifics this round for CTR versus CTS. Faculty level or equivalent, and postdocs, fellows, residents can serve as co-Is. But the mentor has to be, part of the project and from one of the partnering institutions. And a lot of discussion of this. It's a good mentoring experience, mentored training experience for fellows to be co-Is, to be involved in preparation, to be part of a grant. But again, there needs to be sort of that supervisory piece that really is clear as the leads. And PIs, co-PIs, one application per cycle. And Ray, I don't know if you or Sandy or want to speak to it. Is it one application either CTS or CTR per cycle, or potentially one for each? Is there a sense on that?
Well, we would dissuade anyone from
Yeah,
sending the same one to both sides of the fence.
absolutely.
So that will be not, you won't be competitive. So if you have one proposal, select it. If you're going to have two separate and distinct proposals, that may, I mean, you could consider it on either side, but they shouldn't be linked in any way because that would imply you can't do it in the one column without the other column. That will not be competitive.
Right. There is one exception we just want to highlight is that clinicians can be a part of more than one application if it's specifically highlighted there. They are in that sense because they have specific expertise or patient populations that might be relevant to more than one proposal. Needs
We
to
do
be
not discriminate on tenure track or
right.
non-tenure track. That doesn't matter. It's a faculty position.
Right. Thank you for highlighting that too. So hopefully that's clear about the clinical people as well. That I mean that if two scientists have brilliant ideas for looking at a particular set of patients with just cardiac amyloid, for example, and they're both great potential different ideas, different investigators. You may have to work with the same clinician, for example. So in that case, that could be justified, but needs to be really clearly justified. And the clinician themselves, if they know that their patient population is in both, they may want to talk a little bit about why there isn't going to be a competition for patient resources. Letter of intent for both. And we talked about why, and we'll talk a moment more, but required letter of intent, February 20th, 2026 at midnight, forms available in the RFA, listed in the RFA that you received and here. It's a red cap form. Anthony and his team do a brilliant job, and Pam also, in sort of making sure all this gets processed well. And the real purpose is delineated here. It's to ensure a match between the RFA and the project. So, for example, the translational science, we're going to try to make sure that applications submitted as translational science are translational science. And we're not sitting later going, well, it's actually really translational research. and it's not translational science. So that's really an important focus. Now, again, we've had tremendous response to these RFAs, which is wonderful. And the number being awarded is limited. So there is an assessment from the LOI of competitiveness. If there are applications that really don't seem like they're going to be competitive down the road, they are not going to be selected for solicitation of the full application. So it's a brief piece. There's an abstract and a couple other pieces, specific aim, an abstract and a specific aims piece. But so do the best you can to make sure it's clear that these are really competitive projects. The other reason, of course, is to allow us as the leaders and the support staff to begin to identify reviewers of full projects. Institutional sign-off not required for the LOI, and we will do our very best to have a notification in a week, basically about eligibility or not. Okay. I'm not looking at questions. I see chats
Yeah, there's
exploded.
a number of
So,
them, but Sandy's on it and so's
okay,
Anthony.
good.
So let's push on. We've
Yeah.
got only 8 minutes left.
Yeah. Okay, good. Yeah. Full application. We don't actually need to go through this because you can review it if you are then solicited for a full application in terms of what's involved in it, and it's in the RFA. The full application will be due March 13th at midnight. There are three levels of review: an initial administrative review to make sure everything is present that needs to be, an initial review by co-leads program leadership, and decision about the need for additional potential reviewers from our community, as well as potentially from prominent scientists in the field outside of here, and then a final review based on the scores of reviewers and funding decision by program leadership from the alliance partners, the leadership you've seen. And this does involve, again, sort of like at NIH, the reviewers, but the program makes the decision in the end. Review criteria: impact, impact, impact. So prediction of impact. Use of NJAX resources, definitely a plus. We're looking to build collaborations to foster translational research, translational science over the long haul. Obviously, potential generate reproducible findings, the quality of those potential findings and the excellence of the science, potential for translation. Again, the importance of really emphasizing this in the application, likelihood of success. It really can do what you say you're going to do. Valuable to use RWJBA, Barnabas, the health system, for many of the grants, and that would require sign off from them for data. But like all pilot grants, really the potential for generating long legs for the project, publications, funding, adaptation of outcomes is something that really does play an important role.
Yeah, I want to just mention there, Arnold, impact is incredibly important. So project exactly what you're going to do with the money and how is it going to lead to the publication, a timeline for that. When are you going to submit your grants? We count widgets. We count widgets. That means that each and every one of the awardees are tracked, and we characterize the dollar spent against the fold increase that we receive in federal funding or in other funding. That could be industry funding. It could also be foundational funding. So it's very important for us to show a return on investment, a value proposition to NIH that our pilot program fosters X-fold increase. So you know how you get things done when you talk to your boss. The question you ask is, how can I make your job easier?
Right.
Make our job easier by making a lot of money with this pilot, and we all win.
And what I would say, Ray, is that part of the reason that you did so well in the renewal really was that the pilots had an impact, a numerically demonstrable impact. And that helped contribute to the 7 years, I think.
Yep. Thank you.
Institute sign off, we said, may be required for the application itself, depends on institutional policies. and there may need to be separate budgets. That again is important if you get solicited for the full application. For help on whether it fits the RFAs, I'm available, Ray is available, other co-leads are available to try to help understand that. Their RFA has a list of potential co-PIs that could be useful. And again, information on budget subcontracts, you can email NJACTS for that sort of information as well. Our goal is notification of award by the end of April. Actual starting date we talked about depends upon the different components and whether it goes through NCAT's approval and the regulatory approvals. And earliest start date, May 2026. Um, uh, let's see, I think, yeah, we didn't leave a whole lot of time for
Yeah, but
questions,
if
but
you
we
you
had
can take
some.
the slides down and we can
Yeah.
go to
And
the
I'm happy to stay on a little longer if necessary too.
so everyone, let me assure you that Arnold, all the code leads and everyone reviewing these are scientists who are actively writing grants. We take this very seriously. You can see we lean heavily into this program by having unrealistic timelines for ourselves. Because no one wants to waste the time writing a grant and not be competitive. So you're talking to scientists who are going to determine scientifically the merit of your project. We want you to be successful. We want to give out the money to the very best projects. So we are sensitive to that. We will do our very best to give you feedback and to enable a response swiftly so you can move forward. So with that, I want to thank you all. This was, we have an innumerable, we had over 50 for the entire hour. I applaud that. I wish you everybody good luck in this endeavor. Reach out again to Arnold and I. Our emails are very accessible. We're happy to take a quick look to give you as much feedback. We want you to be successful. Arnold?
Yeah, I agree with everything Ray said. I'm not sure that there was an attempt to answer every question, which I really appreciate. I think Sandy and Anthony did a great job. There may be a couple that didn't get fully answered before we stop. So if someone can capture those, maybe we can get back to people as well. That would be good too. So, oh, Sandy, I see you did answer the last one. Thank
Yeah,
you.
no, Sandy,
You were
Sandy
on
and
top of
Anthony,
everything.
they type unbelievably fast, so they can get it answers quickly. So, in all fairness to Arnold and all my colleagues on here, we're going to close this out in 2 minutes. I want to again thank you all. We wish you the very best of luck and happy hunting. So long.
Good luck. Thanks.